Namaste students. Today we will start with the next topic of chapter 20, that is mechanism of muscle contraction. Before starting this topic, let's study about motor unit. What do you mean by motor unit? Because this is important part of mechanism of muscle contraction. So a motor neuron, what do you mean by a motor neuron? Neuron, they are of two types, sensory neuron and motor neuron. The neuron or nerve cell which carry messages from the body part to the central nervous system, that is known as sensory neuron, whereas the neuron which carry messages from the brain or spinal cord, it means from the central nervous system to the effector organ like muscles, that is known as the motor neuron. So a motor neuron, you can see here it is a motor neuron and this is axon of motor neuron. Now the motor neuron, it carry messages from the central nervous system. When this motor neuron, it is connected to a muscle fiber, this is muscle fiber, skeletal muscle fiber. When motor neuron, it is connected to a skeletal muscle fiber, it is known as motor unit. The junction means joining, the region where the skeletal muscle fiber and the axon terminal, they join each other. So the junction between a motor neuron and the sarcolemma. What do you mean by sarcolemma? Plasma membrane of skeletal muscle fiber or skeletal muscle cell is known as sarcolemma. So a junction between motor neuron and the sarcolemma of the muscle fiber is called neuromuscular junction. What it is known as? Neuromuscular junction or motor end plate. Second name is motor end plate. Now here you can see more clearly neuromuscular junction. This is motor unit. Motor unit is, this is the motor neuron and this is the sarcolemma of the muscle cell. Of muscle contraction, it is best explained by the sliding filament theory. This theory states that contraction of a muscle fiber take place by the sliding of thin filament over thick filament. It will be more clear to you later on. Let's start with the mechanism of muscle contraction. First of all, how muscle contraction begins. The muscle contraction, it is initiated by a signal sent by the central nervous system, that is brain or spinal cord, via motor neuron. So the motor neuron, it carries signal to the muscle fiber. So a neural signal reaching the neuromuscular junction releases a neurotransmitter. Here this is the junction, these are the vesicles. So at the neuromuscular junction, the motor neuron, its end plate, its ending, it releases a neurotransmitter known as acetylcholine. What it is known as? Acetylcholine, which generates an action potential in the sarcolemma. What do you mean by action potential? Action potential means change in the permeability of the plasma membrane. Action potential will be more clear in the next chapter 21. So here you should remember that acetylcholine, which is released by the axon ending, it causes action potential in the sarcolemma of the muscle cell. Now this action potential, it spread throughout the muscle cell. I told you earlier that acetylcholine, it changes the permeability of the sarcolemma. When the permeability of the sarcolemma changes, it means the membrane sarcolemma, it becomes excited, due to which the sarcoplasmic reticulum present in the sarcoplasm. What is sarcoplasm? Cytoplasm of muscle cell. So the sarcoplasmic reticulum or the endoplasmic reticulum. The endoplasmic reticulum of muscle cell is known as sarcoplasmic reticulum. So this sarcoplasmic reticulum of the muscle cell, it releases the calcium ion into the sarcoplasm. You can see here the calcium ion, it is released into the sarcoplasm. And what causes the release of calcium ion into the sarcoplasm? That is the neurotransmitter acetylcholine, which has generated action potential, and this action potential, it causes sarcoplasm to release calcium ion into the sarcoplasm. Now, increase in the calcium level in the sarcoplasm leads to the binding of calcium ion with the subunit troponin. Where troponin is present? Yes, troponin subunits, they are present on the actin filament or thin filament. These troponin protein, they have binding site for calcium ion. When the concentration of calcium ion, it increases in the sarcoplasm due to the action potential of the sarcolemma, it causes the increase in calcium level, and this calcium ions, they bind with the troponin protein present on the thin filament and thereby remove the troponin from the active site for myosin. Now, when troponin is removed from the active sites for myosin present on the F-actin, the active site present for myosin on the F-actin are exposed. On the other hand, the myosin filament, that is the thick myofilament, its cross arm. All of you remember cross arm. Cross arm consists of head and short arm. So the cross arm, it has the actin binding site. So the head of the thick myofilament are made up of meromyosin. Okay, so the head of the myosin or thick filament, it binds to the active site, and moreover the ATP, it is converted into ADP with the help of the enzyme ATPase, which is present in the head of the meromyosin filament. More clear with the help of this diagram. Now you can see here, this is the thin filament or actin and this is the thick filament or myosin. Now the myosin head, this is the myosin head. Now when calcium ion, it binds with troponin and removes the troponin from the actin filament and unmasks the active site on actin. After that, ATP, it binds with the myosin head and is converted into ADP and P. P is phosphate and the energy is produced. Now this myosin head, it binds to the myosin binding site present on the actin filament. So you can see in this diagram that myosin head, it binds to the actin filament. This myosin head, it pulls the attached actin filament towards the center of A band. Due to this, the Z line attached to these actins are also pulled inward and thereby causes shortening of sarcomere. This process is known as contraction. We have done in the last diagram that myosin head, they combine with the actin on the myosin head binding site. So you can see here, these are the heavy meromyosin or cross arm. Now these cross arm, when the plasma membrane of the muscle cell, it is under action potential. Then the calcium ion are released. Then troponins are removed from the actin filament and unmask the active site. ATP, it combined with the myosin head, energy is released and it helps in contact of myosin head with the actin filament. These myosin head, they are in contact with thin filament or actin filament. Now, what happens when the head of the myosin, they come in contact with actin filament? So you can see here, it is during the relaxed stage. During the contracting stage, what happened? These more the myosin heads, they are come in contact with the actin filament. When myosin head, they come in contact with the actin filament or they bind with the actin filament, they pull the attached actin filament towards the center of A band. So they move inward, they move towards the center of A band. The Z line attached to these actins are also pulled inward, thereby causing shortening of sarcomere or contraction. It is clear from the above step that during shortening of muscle or you can say during contraction, the I band, which one is I band? This one. So the I band, it get reduced. You can see in the diagram, this is the relaxed stage, this is the contracting, the muscle is under maximum contracting stage. So during this stage, when the contraction take place, the size of the I band, it reduces. So the size of I band, it decreases and these actin filaments, they move towards the A band. This is A band, an isotropic band. So they move inward. You can say they move inward or they may move towards the A band. Whereas A band, it retain its length. So there during contraction of muscle, only the size of I band decreases and you can notice in the diagram the size of A band, it remains the same. So the A band, it retain its length during the contraction of muscle. Now coming back to the last diagram, the muscle has maximum contracted. After that myosin head, it releases ADP and phosphate and the myosin head, it comes back to its original position, means it gets removed. Why? Because it do not have energy for binding to the actin filament. Result in the breakage of the cross bridge. What do you mean by cross bridge? Cross bridge is formed when myosin head, it combines with the actin filament. So when ADP is released, cross bridge is broken, and when again ATP will combine, then again the process will start. So when the breakage of the cross bridge take place between actin filament and myosin head, that result in the relaxation of muscle. During relaxation, it means no neural signal is coming from the central nervous system. So the calcium ions, they are pumped back to the sarcoplasmic reticulum, and where they are stored? These calcium ions, they are stored in the cisternae. Cisternae is the element of endoplasmic reticulum or sarcoplasmic reticulum. And for relaxation, we will move from the downward. When the cross bridge are broken, you can see here all the myosin head, they are combined with the actin filament. Now we are assuming that now the ADP are removed. So the myosin head, they get removed from the actin filament, and then all the myosin head, they get removed from the actin filament, and these actin filament, they slide outward. So during contraction, actin filament they slide inward towards A band, and during relaxation the actin filament they slide outward towards Z line. So now you can see slowly, if we reverse the process, the I band it increases during the relaxation phase, but the A band will remain the same. So when the muscle it return back to its original position, this process is known as relaxation. Repeated activation of muscles will take place and the process will be repeated again. Let's revise mechanism of muscle contraction once again. Central nervous system, it sends the message to the muscle cell via motor neuron in the form of electrical impulses. The junction between motor neuron and the membrane of muscle cell known as sarcolemma is known as neuromuscular junction or motor end plate. Axon ending release neurotransmitter acetylcholine into the synaptic cleft, which combine with the receptors present on the membrane of muscle cell known as sarcolemma, which causes excitation of the membrane or develop action potential, which changes the permeability of the membrane and result in release of calcium ion from the sarcoplasmic reticulum into sarcoplasm. These calcium ion released in sarcoplasm binds with the troponin complex present on the actin filament to unmask the active sites for myosin head binding. Myosin head with ATP combines with actin filament and causes sliding of actin filament towards A band. You can see cross bridge formation between actin filament and myosin head. During muscle contraction, the size of sarcomere decreases. A band it retains its length and size of I band decreases, which result in shortening of sarcomere. You can compare between both the diagram. In one, sarcomere is more in length as compared to the another one. Now, you can see the process. When myosin head it combines with actin filament, ATP is converted into ADP and phosphate. During relaxation, myosin head it gets separate from actin filament, and this process repeat on again. During contraction, myosin head it combine with actin filament in the presence of ATP, and during relaxation, myosin head it gets separated from the actin filament. So, after the separation of actin and myosin filament during relaxation, calcium ion removed from troponin complex and returned back to sarcoplasmic reticulum, and the size of I band increases.